students, BioWorld welcomes you to another exciting new session. Thank you so much for the comments and feedback of the previous sessions. From that itself, I could make out that you people are very much excited for the next session. Am I right? Today, we are moving into the next chapter of the second volume text, Genetics of the Future. I am quite sure that you will enjoy this chapter very much. Why? Because it deals with the genetic engineering principles, where most of the fictions that we have read in our books will come into reality in the near future. So shall we begin?
History of biotechnology. Humans have used biotechnology since the dawn of civilization. Egyptians used yeast to bake leavened bread. What do you mean by leavened bread? Leavened bread contains some rising agents, for example yeast, to make the dough to bubble and rise and create a light, airy product. The Chinese developed fermentation techniques for brewing and cheese making. The Aztecs used spirulina algae to make cakes. Here you can see the ancient technology of winemaking. The cheese production, one of the most famous and oldest cheese is Parmigiano Reggiano, commonly known as Parmesan cheese. The monks living in Italy created this cheese when they needed to find a way to extend the shelf life of large quantity of milk they were producing. Ancient bread industry. Today, in the modern life also, we make use of microbes for processing food and other purposes. Example, appam for making wines, breads, leather industry, where we make use of a bacteria called Bacillus subtilis. This microbe is used in pre-tanning processes, that is in soaking, dehairing, baiting and degreasing of the leather to produce wide variety of products from the leather. Who invented biotechnology? Hungarian engineer Karl Ereky first coined the term biotechnology in 1919, meaning the production of products from raw materials with the aid of living organism. So with the help of small living organisms, certain products are produced, and the term was first coined by the Hungarian engineer named Karl Ereky. Genetic engineering, the modern version of biotechnology. Genetic engineering is the technology of controlling traits of organisms by bringing about desirable changes in genetic constitution of an organism. The basis of this is the discovery of the fact that genes can be cut and joined. So in a simple way we can say that the genes can be manipulated to cause desirable changes. Genetic engineering is the modern form of biotechnology. Paul Berg is considered as the father of genetic engineering for his valuable contribution in this field. Principles of genetic engineering. Children, what is this? Yes, very good. Scissors. We use scissors for cutting. So the first principle is to cut the genes. Second one, copying. That means copying the desired genes. And finally, the glue. For what purpose the glue is using? Yes, correct. It is for joining. That means for pasting. So the three basic principles of genetic engineering are cut, copy and paste. The desired genes are cut or the unwanted genes are cut and removed. The desired genes are copied and it is pasted or ligated. Now let's have a look on the tools which is used in genetic engineering. The major tools include genetic scissors, that is used for cutting the genes, genetic glues for ligating or for attaching or joining, vectors, the agents or the organisms which are acting as a mediator for all these processes, and the desired gene. So genetic scissors, genetic glues, vectors and desired genes are the major tools used in genetic engineering. Let's have look on each one in detail. Genetic scissors. As I mentioned, the scissors indicates for cutting. For cutting the genes, we use certain enzyme. They are commonly known as genetic scissors. Example is restriction endonuclease. Restriction endonuclease. Next one is genetic glue. The glue which we make use of joining the segments together. Example ligase. Here you can see an animation in which the DNA segments are joined with the help of the ligase so that a recombinant DNA, that is new combination of gene or DNA, is made possible. So let's see how are the very minute genes are cut and joined. The enzyme used to cut genes are called genetic scissors. Example, restriction endonucleases. The enzyme used to join DNA or the genes are called genetic glues. Example, ligase. Now let's move to vectors. A gene from one cell is transferred to another cell by using suitable vectors. Vectors which contain the ligated genes enter target cell. Plasmid in bacteria are generally used as vectors. In this way, new genes become a part of genetic constitution of the target cell. So vectors play a crucial role in this process. Plasmid. As you know, bacteria is a prokaryotic cell without a definite nucleus. In addition to the normal bacterial chromosome, they have an extra chromosomal structure called plasmid, which is usually circular in shape. And they are very easy to manipulate, that means to cut and join. So this plasmid is used as a tool in genetic engineering. In this animation, you can see the plasmid gene is cut and removed, and in the place the desired gene is joined. So here you can see the desired gene is joined, and again this plasmid is inserted into the bacterial cell. As the bacteria multiplicates, the gene also multiply into number of copies which can be isolated into different products. Scope of genetic engineering. Gene editing, gene therapy, application in medicine, human genome project, DNA fingerprinting, genetically modified animals and plants, and for controlling pollution. So genetic engineering has plethora of applications in the modern era. So let's have a look on each one in detail. First of all, gene editing. Today, genetic engineering can edit the genes in the genetic constitution of organism just as editing an essay. It's quite interesting, isn't it? The most modern aspect of genetic engineering is gene editing. CRISPR-Cas9 is one of the most effective scissors used for this process. This contains Cas9, an enzyme, and a guide RNA, that is gRNA. Active researches on gene editing are being conducted worldwide. Next one is gene therapy. The gene editing technology has created an avenue to the endless scope of gene therapy. It is a method of treating genetic diseases. Defective genes are replaced by the functional genes. In the illustration you can see the defective genes are replaced by the corrected genes. In this way we can treat the genetic diseases. The next application of genetic engineering is in the field of medicine. Many proteins that can be used for treatment of diseases in humans are produced through genetic engineering. The various proteins used for the treatments are tabulated here. Interferons, which are widely used for treating viral diseases. Insulin, which is quite common protein produced as a result of genetic engineering, is used for treating diabetes. Endorphin, used as a pain relieving agent, and somatotropin is used for treating growth disorders. So all these can be easily produced through genetic engineering technology. Here the illustration depicts the formation or the isolation of insulin from bacteria through genetic engineering technology. So from the human cell, from the human DNA, the insulin gene is cut and removed, and it is inserted or joined in the circular DNA of the bacterium, that is the plasmid. So now the bacterium plasmid have the gene of human insulin. The plasmid with ligated insulin gene is inserted into the bacterial cell. Bacteria that multiply in culture medium produce inactive insulin. So this active insulin is produced from the biotechnology or the genetic engineering process, is commercialized and is used in the form of medicine. Human Genome Project. A project to reveal the secrets of human genome. What do you mean by genome? The complete genetic material in an organism is called genome. Gene mapping is the technology that helps to identify the location of a gene in the DNA which are responsible for a particular trait. So if we identify the typical gene connected with the trait, we can easily rectify the defects. The non-functional genes that code for proteins are called junk genes. Why the Human Genome Project is so important? The project completed in 2003, which gives us the ability for the first time to read nature's complete genetic blueprint for building a human being. All the traits are encoded in the form of gene. So secrets revealed are Human genome has about 24,000 functional genes. Major share of human DNA includes junk genes, which are non-functional. There is only 0.2% difference in DNA among humans. About 200 genes in human genome are identical to those in bacteria. So we have a relation with bacterial cell too, which indicates the similarity of around 200 genes in human as well as in bacteria. Now let's explore more applications of genetic engineering. Farm animals. One of the future promises of genetic engineering is the farm animals. Desired genes are inserted into animals and transform them into farm animals. Genes responsible for the production of insulin and growth hormones required for humans are inserted into animals like cow, pig, etc., transforming them into farm animals. There are certain limitations in producing insulin using bacteria. As you know, bacteria are microscopic in nature and it's very tough to manage them in a sterile condition. So the most important hurdle in this field is culturing of bacteria. Researchers in this field show that instead of this, medicines can be extracted from the blood or milk of genetically modified animals. So such animals are known as farm animals. We make use of genetic engineering in poultry too for obtaining superior quality and quantity products. Genetically modified salmons. Salmons, as you know, it's a type of fish. Here you can see the difference between a regular salmon and genetically modified salmon. Genetically modified mice, which is widely used in researches in the field of medicine and in organ transplantation. Here you can see a small ear is growing on human ear is growing on the back of the mice, which can be used for transplantation. This is another picture showing human ear growing on the mice. GM glow fishes. The glowing organisms under UV light have paved way to the break researchers in medical field, especially for treating cancer, Parkinson's and spinal cord injuries. Glow in the dark cats are also used for HIV researchers. So not only for ornamental purposes, they have wide variety of uses in researchers and medical field. Next one is spider goat. Spider goat is a great example of genetically modified organism. They aren't scary as they sound. Goats have had the spider-like gene inserted into the DNA, so when the female goat lactate, reduce breast milk, the milk that contains spider silk, which is pretty incredible. It is very strong as steel and so light and long strands are obtained, which is very unique material having thousands of applications. Let's explore genetically modified plants in agriculture. We call them as genetically modified crops. In order to get superior quality of fruits, vegetables and so on, genetic engineering is used in agriculture too. Bt plants. Bt stands for a bacteria called Bacillus thuringiensis. This Bacillus thuringiensis have the ability to kill the insect. So once the bacteria get inside the insect body, the intestine of the insect get destroyed, and as a result it causes the destruction of the pest. So this bacteria is incorporated in the plants and make use of pest resistant plants. So here you can see Bacillus thuringiensis, the gene is isolated and incorporated in a crop plant. The Bt gene is inserted into crop and the crop is infected by a European cone borer, a type of caterpillar, and the pest dies when feeding on any part of the plant. Similarly, Bt brinjal, Bt cotton, Bt maize, and wide variety of fruits and vegetables can be produced. One of the amazing product formed as a result of genetic engineering is tomato. Doesn't this term sound a little weird? You might be wondering whether it is a tomato or potato plant. Actually both. Yes, you heard me right. In this single plant, tomato grow above the ground while potatoes grow below the ground level. And how is this made possible? This can be done through the process called grafting. In this, the stems of the both tomato and potato are chosen and tied up together to form a single plant. This is an example for plant engineering. Quite interesting, isn't it? Another example is genetically modified potatoes, which we usually use for making French fries or long potato chips. Golden rice, which contain not only carbohydrate but also vitamin A. Flavor saver tomatoes, which having a more shelf life than the normal tomatoes. Next application of genetic engineering is DNA fingerprinting. As you know, fingerprint is unique. Similarly, the arrangement of DNA is also unique in each person. It was Alec Jeffrey who invented this technology. Here you can see the DNA obtained from the crime scene as well as 3 suspects' DNA. When we compare the samples with the crime scene, you can easily identify that suspect 2 has more similarity with the crime scene DNA. From this, we can conclude that suspect 2 is related with the crime scene. So in this way, from the DNA sample, we can easily prove the crime cases and various disputes. So let's have a look on DNA profiling. The technology of testing the arrangement of nucleotide is called DNA profiling. The DNA profiling is unique in each person. From where the DNA samples can be collected from the skin, hair, nails, blood and other body fluids. Skin, hair, nail, blood and body fluids. Now let's move to the applications of DNA fingerprinting. To find out hereditary characteristics, to identify the real parents in cases of parental dispute, to identify person found after long periods of missing, to identify the real culprit of crimes, robbery, etc. The next application of genetic engineering is related to controlling pollution. Superbugs, a type of bacteria developed as a result of genetic engineering, is used for controlling oil pollution. So for treating oil spills and thereby controlling oil pollution, we make use of superbugs. It's a type of bacteria which is artificially developed. AE104, another microbe which is used for removing heavy metals from effluents. In addition to that, we also make use of microbes in wastewater treatments and sewage treatment plants. Is genetic engineering a boon or curse? Let's have a look on this collage. Threat to indigenous variety. What do you mean by indigenous variety? Native or the local varieties. It is criticized that genetically modified varieties cause harm to indigenous varieties and may cause health issues to human. So gradually it caused the extinction of natural or the native or wild varieties. Bioweapons, a new challenge. Application of genetically modified pathogens and pathogens multiplied through biotechnology upon enemies is called biowar. This becomes a threat to the existence of human beings. Genetic modification, violation of rights. Certain organizations argue that genetic modification is an intrusion upon the freedom of living beings and it's a violation of rights. So admittedly, genetic engineering is a mixed bag of boons and banes. Science and technology are the products of man's reasoning ability. We can justify this only if they are utilized for human benefit.
So children, what we have discussed today? Yes, the history of biotechnology, the principles of genetic engineering, and plethora of applications of genetic engineering which we are widely make use of. So here I am winding up. Thank you.